Hello students, welcome back to SRM Biology classes. Today's topic is with reference to the plasmids. The term plasmid was discovered by William Hay and Josh Lederberg in the year 1952. In case of prokaryotic bacteria, they discovered first plasmid. In invalid terminology, the plasmid is defined as it is an extracircular double-stranded DNA. Exhibits self-replication or it exhibits autonomous replication. It is found in the bacterial cytoplasm. That extracircular double-stranded DNA that replicates autonomously in the bacterial cytoplasm. Is called plasmid. If you observe the structure of the bacteria, you can find the presence of two types of DNA. The first one is linear DNA. This linear DNA will take part in inheritance and variation. In addition to the linear DNA, you can find the presence of extracircular double-stranded DNA that will replicate autonomously in the bacterial cytoplasm. That unique extracircular double-stranded DNA is called plasmid. The term plasmid was discovered by William Hay and Lederberg in the year 1952. Today, the plasmids are widely used as a cloning vector, vehicle used to transfer genetic material from one organism to another organism. Today, the plasmids from the bacterial origin are widely used in the recombinant DNA technology in order to manipulate the genetic makeup of the living organism. Today, the plasmids are widely used as a cloning vectors in all the techniques of gene manipulation. Plasmids exhibit great degree of diversity. The plasmids exhibit great degree of diversity. Their size, it may ranging from 2000 base pairs to 1 lakh base pairs. The number of plasmids in the bacteria is also highly variable in number. Its number ranging from 1 to 20. Its number is highly variable in case of bacteria and its number ranging from 1 to 20. Some of the classical examples for plasmids are PUC-18 and the pBR-322 are some of the important types of plasmids widely employed in all the techniques of gene manipulation. The plasmid is defined as extracircular double-stranded DNA exhibits autonomous replication, and it is found in the bacterial cytoplasms. That such extracircular double-stranded DNA is called plasmid. Today, wide variety of plasmids are widely used in the gene manipulation or widely employed in the gene manipulation. With reference to the important type of plasmid, from the point of NCERT curriculum, you are having the main important structural details of a plasmid called pBR-322. Plasmid of Bolivar, P refers to plasmid, B refers to Bolivar and Rodriguez. pBR-322. 322 is the strain number, is the bacterial strain number. This unique type of pBR-322 is normally found in the, and it is naturally found in the E. coli bacteria. It is naturally found in the E. coli bacteria. With reference to the main important structure of pBr-322 plasmid, P refers to plasmid, B refers to Bolivar, R refers to Roderick, and its strain number is 322, and its strain number is 322. With reference to the main important introductory aspects of the pBr-322 plasmid, it is a naturally occurring plasmid found in an nanobacteria. The name of the nanobacteria is E. coli, Escherichia coli. The human intestine is characterized by the presence of a unique nanobacteria called E. coli. From that E. coli bacteria, a naturally occurring plasmid was discovered. The name of the plasmid is pBr-322. With reference to the main important structural details of the pBr-322 plasmid, it is characterized by the presence of a size of about 4.3 kilo base pairs. 4.3 kilo base pairs. It is characterized by the presence of 43,000. It is scattered by the presence of 4,363 base pairs. 4,363 base pairs. With reference to the main important structural details of the pBr-322, it is characterized by the presence of Warite site. It is a type of extracircular double-stranded DNA. Extracircular double-stranded DNA. It is characterized by the presence of Warite site. Warite refers to origin of replication. It is the site where the replication starts. In addition to this, any piece of DNA is linked to the plasmid, and it is made to replicate autonomously in the host cell. Such a unique region is called Warite site. It is the origin of replication. It is the region where the replication starts. Any piece of foreign DNA to be linked to the plasmid and allowed to replicate in the host cell, within the host cell. Such a unique region is called Warite site. If you observe the structure of the pBr-322, it is characterized by the presence of two selectable markers. The two selectable markers are namely ampicillin resistant genes, and the second important selectable marker is the tetracycline resistant gene. The ampicillin and tetracycline resistant gene are the two important selectable markers found in case of pBr-322. The first important selectable marker is called ampicillin resistant gene. The ampicillin resistant gene, it is characterized by the presence of two recognition sites. The two recognition sites are namely, the first important one is PST1 and the second one is called PVU1. PVU1 are the two important recognition sites found on the ampicillin resistant gene. But in case of tetracycline resistant gene, the tetracycline resistant gene is the another important selectable marker. It is characterized by the presence of two recognition sites. The two important recognition sites are namely SAL1 and BAMH1. BAMH1 are the two important recognition sites naturally found on the tetracycline genes. These selectable markers, namely ampicillin and tetracycline resistant genes, they helps in identifying and eliminating. They take part in identification and elimination of non-transformates and permitting the growth of transformates in the plasmid. They play a very important role in identifying and eliminating non-transformates and permitting the growth of transformants. These genes encodes resistance to antibiotics, namely ampicillin and tetracycline. These genes encoding resistance towards two important antibiotics, namely ampicillin and tetracycline. In addition to this, it is characterized by the presence of 13 unique restriction sites. It is characterized by the presence of 13 restriction sites. There are several restriction sites in case of pBR-322. Out of 13, 7 are the important restriction sites. 7 are the important restriction sites. The restriction site is a unique site. It is characterized by the presence of specific restriction endonuclease recognition sites. Restriction endonuclease. It will provide the place for the action of restriction endonuclease. The enzyme restriction endonuclease is popularly designated as molecular scissor. Molecular scissor or molecular knife. If you observe the 7 important restriction sites, the 7 important restriction sites is the unique site in which you can find the presence of restriction endonuclease recognition sites for the action of several types of restriction endonuclease enzymes, namely E Coli. E Coli. In that particular position, the enzyme E Coli will be acted in which the foreign gene is to be inserted. Particularly in this region, the plasmid is subjected to cut. In that cut region, the foreign gene is to be inserted very precisely. Opposite to the recognition site, you can find the presence of ROP region. It is the region where it will code for the protein involved in the replication of the plasmid is found in the ROP region, or refers to replication of plasmid. ROP refers to replication of plasmid, particularly on that side. It take part in the action of a protein that normally initiates the DNA replication. Such a unique regions are normally found in a unique plasmid called pBR-322. This is with reference to the main important structural details of the plasmid called pBR-322. The next important aspect is with reference to what are the main important competent hosts widely used in the gene manipulation and also in the recombinant DNA technology. The competent host is defined as the cell in which is capable of taking foreign DNA. The cell in which is capable of taking foreign DNA is termed as competent hosts. The competent hosts are required for the transformation of recombinant DNA. Universally, the DNA is hydrophilic in nature. It shows high degree of affinity towards the water. Because of this unique characteristic feature, the water is purely designated as liquid of life. It is because of the hydrophilic nature. It cannot pass through the cell membrane, because the cell membrane is lipophosphorescent in nature. It is scattered by the presence of lipid residues. Because of the hydrophilic nature of the DNA, it cannot pass through the cell membrane. Thus, it requires host cell. One of the widely used host cell is the bacteria, and it is made competent to take up the foreign DNA. Such a unique one is called competent host. The cell which is capable of taking the foreign DNA is called competent host. One of the widely used competent host in the recombinant DNA technology in order to take up the foreign DNA is the E. coli. The origin of E. coli, it will date back to 4.5 billion years. 90% of the human genetic material and 90% of the E. coli genetic material is similar in nature. With this interpretation, it is very clearly illustrated that from the prokaryotic bacteria led to the origin of eukaryotic human beings on the surface. Today, because of the enteric nature of the E. coli, in addition to this, it is characterized by the presence of some unique characteristic features. The E. coli is widely used in the genetic engineering experiments in gene manipulation, or in order to uptake the foreign DNA. Some of the important salient features of the E. coli as an competent host are namely, the E. coli is non-pathogenic in nature. It is the part and parcel of the human enteric system. It is very easily culturable under laboratory conditions. In addition to this, it is scattered by the presence of very less generative period. Within 19 minutes, it will successfully complete one life cycle. Because of this unique nature, we can get the results very quickly. In addition to this, it exhibits transformation process. With the transformation process, it effectively transfers genetic material from one organism to another organism. In addition to this, from the point of genetic engineering experiments, it is characterized by the presence of plasmid. In that plasmid, we can effectively incorporate the foreign gene. In addition to this, the plasmids have limited number of genes. In addition to this, the handling of the E. coli bacteria is very easy. In addition to this, we can get quick results under laboratory conditions. Because of all these unique characteristic features, the E. coli is widely used as one of the competent hosts in the recombinant DNA technology. There are 4 methods are widely employed in the recombinant DNA technology to introduce the foreign DNA into the competent host. the four widely employed methodologies to introduce the foreign DNA into the competent host cells are namely, the first one is calcium chloride and heat treatment. The second important one is the micro-injection process. The third one is biolistic or gene gun methodology, and the last one is disarm. It is non-pathogenic. Disarm refers to non-pathogenic organisms are widely employed in order to introduce the foreign DNA into the competent host. The first important methodology is called the calcium chloride and heat treatment. In this method, the host cell is treated with calcium chloride. Because of calcium chloride nature, it exhibits the thinning of the bacterial cell wall is resulting in the loss of permeability, followed by the sudden heat shock at a temperature of about 42 degrees Celsius. Later, after heat shock at a temperature of about 42 degrees Celsius, it is subjected to cold treatment. By placing on an ice pack. This results in the depolarization of the cell membrane. When bacteria is treated with calcium chloride, resulting in the sudden loss in the permeability, followed by the heat shock at a temperature of about 42 degrees Celsius. Once the heat shock is done, it is subjected to cold treatment by placing them on the cold pack. When it is subjected to cold pack, it results in the depolarization of the cell membrane. This creates the formation of large pores in the cell membrane. The presence of large pores in the cell wall which make the host cell to uptake the recombinant DNA very easily, which can uptake the host cell to take up the recombinant DNA very easily. Such a unique method is called calcium chloride and heat treatment. The second important methodology is micro-injection. In this method, the recombinant DNA is directly introduced by making use of micro-injection. The recombinant DNA is directly injected into the host by making use of microsyringe or by making use of micropipette. Is the another important microinjection methodology in order to introduce recombinant DNA into the host cell. The third one is the gene gun or the biolistic method. In this method, the cells are continuously bombarded with high velocity of either tungsten or gold particles. These gold and tungsten particles are imprinted with desired DNA This method is widely employed in order to introduce the plant genes into the host organism. The biolistic or gene gun method is widely used. In this case, by making use of gold and tungsten particles, high bombardment is done with great velocity. On the tungsten and on the gold surface, you can find the impingement of the disarmed DNA. When gold particles imprinted with the desired DNA, if you observe the hide and seek, you can find the impingement of chocolate bits in the biscuit, like that. On the surface of the tungsten and on the gold particles, desired recombinant DNA is imprinted with a great velocity. The tungsten and the gold particles are bombarded onto the host cell, resulting in the incorporation of recombinant DNA into the host cell. This biolistic or the gene gun methodology is widely employed in order to introduce recombinant DNA into the plant cells. The biolistic or the gene gun methodology is widely used. The last important methodology to introduce recombinant DNA into the host is disarm pathogen. The disarm pathogen refers to the use of non-pathogenic virus as an vectors to infect the host cell. As a result of infection, it effectively transfers recombinant DNA into the host cell. In this methodology, the disarm retroviruses are widely used as a disarm pathogen. These retrovirus vectors are allowed to infect the host cell. When retrovirus started to infect the host cell, resulting in transfer of recombinant DNA into the host cell. This methodology is ideal for animal cells. These are the 4 important methodologies widely used to introduce foreign DNA into the competent host. The last important aspect is bioreactor. That will be studied in the next class. Thank you very much.